Okay, so my name is Tara H. Boyer. I did my research on the effects of irrigation levels on pests, floral display, nectar quality, and imidacloprid uptake in salvia under my faculty mentor, Dr. Erin Wilson Rankin. And I did graduate in March 2021 with a bachelor's of science in biology. So for my background, I wanted to go over what everything is because that title is quite long and you guys might be wondering what each thing actually is. So for the experiment, we used a hybrid sage, which was Salvia clevendale crossed with Salvia leucophylla, which is Cleveland sage and San Luis Obispo purple sage. They are both native to Southern California with some overlapping range. Specifically, Cleveland sage is found in more of the coastal regions of Southern California into Baja California, and they prefer elevations under 900 meters. In contrast, Slow's purple sage is more of the mountainous sage of Southern California. And then moving on to imidacloprid, it is a popular commercialized neonicotinoid pesticide that has been on the market since 1994. It is used all the way up into agriculture, down to nurseries and into people's backyards. And it actually targets the insect's nervous system. So if you look at the word neonicotinoid, you can actually see that nicotine is in it. So it targets the nicotine receptors and essentially poisons the insect over time. So I also go into label rate since imidacloprid has a label rate. For now, we're just going to say it's similar to a serving size of that you see on packages of food. And irrigation is the amount of water that affects the plant's quality of life. If you think about it, plants are unique. So some plants do prefer to have more water than others. So if you think tropical plants are more water thirsty, while cacti are more like, don't water me that much. So it just depends on the plant you have. So for our experiment, sage are a drought tolerant species. If you remember, we do live in Southern California, so we don't get a lot of rainfall here. So it makes sense that sage have adapted to not require as much water as previously. And finally, we have pests. So pests decrease the plant quality and markability. If you go to a nursery and you're looking at a plant to buy, you're not going to buy the plant that's covered in aphids or has holes in it from insects chewing on it. And it's the same sort of thing when we're buying fruit and vegetables as well. So it's really common for pests to be controlled with pesticides. And I wanted to quickly show what salvia sage looked like in our greenhouse at UCR's campus. As you can see, it's a nice little green shrubbery plant with these magnificent purple flowers. And salvia are popular among pollinators. So this is a carpenter bee on some salvia. So irrigation and pesticide application are two management methods affecting plant health. So how exactly do water levels affect the production of nectar and thus the uptake of imidacloprid as well? And so this question became quite complex. So to show a better representation, we created a graphical abstract just to break down each part of the experiment and show it in a more cohesive manner that makes more sense. So I'll start over here in the pesticide section with imidacloprid. So we did have 4 levels of imidacloprid applied depending on the label rate. So for the white square over here, this was actually our control. So no imidacloprid was applied. And moving all the way over to the red, this was the 1/3 label rate. So for our experiment, we did not apply the full label rate that was specified on the bottle of imidacloprid. We applied 1/3 of that. And so to give a visualization, let's say the label rate for the imidacloprid was 1 tablespoon. We only applied 1/3 of that tablespoon. And then as you decrease down, this is the 1/30th and the 1/300th. Moving into the water treatment, we had low and high water together, and then these two interacted, which influenced our 2 pests in the experiment. And then the pests influenced exactly how the plant did and how the pollinators do with nectar and that sort of thing. So for morphology, it gets quite complex again with the plant parts. We had bud, floret, and inflorescence. So bud is the pre-budding stage of the flower, as you can see with the arrow here. It's getting the flower ready to bloom, all that sorts of things, but it is not flowering yet. While a floret is a single flower in this stage here, and the inflorescence is all 7 of these flowers present. So as you can see, you can have 7 florets for 1 inflorescence. And once this bud starts transforming into florets, you'll have a second inflorescence on this plant. So Brix percentage is the sugar concentration of nectar and the amount of sugar found within a water or aqueous solution, while nectar volume is just the amount of nectar found within a single floret. And then I'm going to describe the pests a little bit. So we have scale insects. They are mostly immobile and they like to hang out on the plant, specifically on the stem and the leaves, and their mouth part pierces through the plant cell's wall and sucks up the nutrients passing by. In contrast, thrips actually commonly aggregate inside florets, so they are found inside the actual floret. They are not on the outside of the plant. And they actually feed on the plant's outermost layer of plant cell wall and the chloroplast-rich cells. So I wanted to quickly demonstrate that we did have a difference in our water treatments with low and high water. As you can see, this 3 stars represent that the data was strongly significant. And over here on the y-axis, we have the soil volumetric water content. And on the x-axis, we have water treatment. So you can see that the low water is around 0.15 for soil volumetric water. In contrast, the high water is closer to 0.2. So we did have variation with each plant, as you can see with the dots that continue up. But overall, the majority of the data for either treatment did fall within these ranges and signified that we did have a low water treatment and a high water treatment. And I'm going to move into our 3-way interactions now. So this figure might be a little daunting because it is a 5-paneled figure, but we did this because pesticide, water, and pest interactions had a monumental effect on our experiment. And you're looking at each floral life stage. So you're looking at number of buds, florets, inflorescence, nectar volume, and Brix. So the reason why it looks overwhelming is because we have all of these life stages, but I'll go through each life stage with you guys individually and explain them. And the reason why we look at the life stage is each life stage influences how the plant does. So if you have a certain number of buds, that will mean you'll have a certain number of florets, which means you have a certain number of inflorescence, which means you'll have a certain amount of nectar. So for buds, I want to point out our pesticide key is up here in this corner, and the circle with black filled in is the 1/3 treatment, with decreasing all the way down to the control with a green open circle. And over here, we have the number of buds treatment. We have low and high water treatments. And then we have pests absent, pests present. And then this is for scale, and this one's for thrips. So as you can see, buds increased with high water and presence of thrips. If you look at the graph itself, you can see over here that this thrips and high water is clearly demonstrating what's previously said above. And the low water isn't just having an effect as much. And you can clearly see the difference with the high and the high here. So if you have buds increasing, that means that you should be in a high water treatment, which means you also should have thrips present. So moving on into inflorescence, for the simplicity of the symposium and the time frame, I'm not going to go over florets with you guys, but it is important to note that inflorescence and florets had very similar data. So that's why we took out the florets, and I just preferred talking about the inflorescence. So plants with more inflorescence actually have more thrips. And you can clearly see this once again on the graph, especially in the high water treatment. And the low water is still pretty significant as well. You can clearly see how for the low and high, when the thrips are absent, they're all around 0, but you get drastic increases with thrips present in here. And so moving on to Brix and nectar volume, there were no major effects of treatments overall. It is important to note we did have effects on certain treatments, specifically, let's say like the 1/3 treatment, high water and scale would have effect, but it wasn't as consistent and reliable as the previous data on this other slide. And so overall, there were no major effects of treatments. In certain treatments that we specify into, there were effects, but overall, it just was not consistent enough for us. And so I want to conclude with pesticide treatments for the results we had in the experiment. So as you can see, once again, We have stars signifying significance, while NS means not significant. We have imidacloprid parts per billion on the Y, and on the X, we have the pesticide treatments, along with the light blue signifying low water treatments and dark blue signifying high water treatments. So for the control in the 1/30th pesticide treatment, you can see that the data was not significant. They are two different treatments, but when we analyzed the statistics, the data signified that they were actually essentially the same treatment. You can see that the 1/300th does have a slightly higher amount of imidacloprid than the control. However, statistically speaking, they were the same treatment. But looking at those two compared to the 1/30th, you can see there is an increase of imidacloprid, specifically with the low water having a higher amount than the high water. And then moving on into the 1/3 treatment as well, you have a drastic uptake of imidacloprid in the plant, once again following the pattern with the low water having more than the high water. And I just want to note that this red line signifies lethal levels of imidacloprid for pollinators. We ourselves did not test on any pollinators for our experiment. This is just previous literature that has been published signifying lethal levels are around the red line. So for our conclusions, every life stage was affected by pesticide, water, and thrips interactions, as I went over with you guys. Scale did affect some life stages, but it just wasn't as common as thrips. And this possibly could be because thrips are found inside those florets and scale are just more superficial. Water and pesticide alone did not influence the interactions, but pesticide and water had a strong relationship. If we think back to the buds, if that high water treatment produced more buds, which meant thrips were present. And then we did mention that bricks and nectar volume were the only life stages not affected, which was a little surprising, but not surprising at the same time. But as I mentioned, pesticide analysis showed lethal doses of imidacloprid in nectar for pollinators. I want to reiterate that we did not test on pollinators, so we can't for sure know that our value was lethal, but previous literature has said that the lethal levels we found were in there, especially for the 1/3. The 1/3 treatment was really high. And the 1/30th was more around the lethal levels, especially I believe the high water treatment was a little bit lower. So this is interesting too, because we applied the imidacloprid 12+ weeks after the pesticide analysis. So ingestion even 12+ weeks after we applied it could really kill pollinators. And that is huge because you're applying this in backyards and in nurseries. Nurseries are a really common area for pollinators to come and visit. So those are floral sources they need. And with lethal levels of imidacloprid that we found, that does cause an issue. And I want to note that imidacloprid may actually be an ineffective pesticide for pests, specifically against scale and thrip insects. In our experiment, these both were present before the application, during the application, and after the application, and they did not seem affected at all. They continued moving on with their lives and reproducing and just continuing on, which is very interesting because imidacloprid is used to manage those pests. So the fact that scale and thrips did not seem to be affected is monumental. And finally, water treatment. Higher water treatment supported less uptake of imidacloprid in comparison to low water treatment, which makes sense because if you're watering a plant more, they're going to take up the water more essentially. And water heavily influenced the pest interactions as well, as I mentioned before, with the buds and the high water. And then I have some acknowledgements. Special thanks to Dr. Erin Wilson-Rankin. She was my faculty mentor and co-author on this. We sat through many Zoom meetings because our studio isn't the most user-friendly. And we talked a lot about the paper, the research, everything going on. And I just wanted to thank her for all the help. I also wanted to thank Dr. Jake Cisalia. He just graduated like 2 days ago with his PhD in entomology. So congratulations to Jake. He helped a lot with setting up the experiment, picking up the plants, organizing the plants, applying the treatments, and so much more. And then I also wanted to thank David Rankin. When COVID hit, I wasn't able to collect a lot of the data. So Dr. Rankin and David also really stepped up and were able to collect a lot of the data for me. And he provided a lot of the pictures with the pollinators in the stage as well. And any questions?